everyone. Yeah, so I'm Antonia Lock. I'm a curator here at Uniprot at EMBL-EBI in the protein function content team. And I've also got Paul Denny here, who's helping me answer questions that are being asked throughout the webinar. So the objectives of this webinar is to explain what the gene ontology, or the GO, is, and what some of its limitations are. I'm going to explain where the different GO annotations come from, and various methods of accessing GO annotations, and also a little bit about how GO can be used in analyses. So the gene ontology, the different people who contribute to the gene ontology comprise the GO consortium. And it's a number of various and diverse annotation groups. I've got a link in the presentation there. The slides should be available after the webinar. So if you want these links, you can get it from there. Yeah, so it's a number of very diverse different annotation groups. So there's model organism databases, there's Uniprot, and annotation groups with other focuses like for example, those that focus on genes involved in human disease and so on. And the gene ontology, it aims to integrate data about gene product function. And I'm going to use gene product and the term gene a little bit interchangeably, but when I say gene, I actually mean gene product. So for example, proteins and non-coding RNAs. So the GO integrates data about gene product function across a very diverse range of organisms, ranging from bacteria to human. And what the GO does, it allows the transfer of knowledge from model organisms to humans, for instance, or between each other. And it also enables computational analyses to interpret high throughput experimental and clinical data. And of course, because the traditional method of disseminating scientific findings, so via publications, is sort of a very low throughput method of sharing knowledge, whereas the gene ontology allows sharing of data at a more efficient scale. And the GO consists of two parts. So you have the ontology itself, and also the annotations that are made to the ontology. So basically what an ontology is, it's a dictionary or a list of terms, but it's not a simple flat list of terms. They're actually connected to each other via different types of relationships. And what an annotation is, it's when you associate a gene product, protein or non-coding RNA, to an ontology term. And the gene ontology is freely available. There are different companies that will offer certain services, perhaps, but the underlying data is in charge for. And more specifically, the aims of the GO ontology project is to capture biological knowledge and describe different attributes of gene products in a species agnostic manner. So it shouldn't matter. You should be able to use the same terms if you talk about a bacterial protein as if you're talking about a mammalian one. And it also tries to use a language that is both understandable by humans, but is also computable. And therefore it provides a resource that's suitable for analyzing high throughput data or large scale data sets, not necessarily high throughput. And what the GO is not, it only describes normal processes and functions. So it doesn't cover various pathological processes, such as those that happen in disease, or artificial ones that might just happen in vitro. And GO is also not static. So both the ontology and annotations are reviewed and updated pretty much on a daily basis. And you can see that here, it's not so important what's on the different axes, but this is basically showing the number of annotations to human over the last couple of years. And the annotations have increased from about 500,000 to over 600,000 in the Gene Ontology database. So that shows you if you use a data set that's just a few years out of date, you will lose out on a large amount of annotations. And similarly, the GO ontology is updated as well. So terms may be obsoleted, which are shown here in orange. They might be merged with each other, which is shown in red here, and new terms in blue are also created. So it's a it's a very organic thing, I suppose. And it's not only growing, it can also be shrinking a little bit. So I know for the last few years, there's been a lot of work done on pruning out overly detailed terms in the ontology that should be captured in new ways instead. So for example, the number of, so Go comprises 3 different aspects, which I'll go into more detail later on. And number of process terms, for instance, has actually been decreasing over the last few years. And there's lots more stats about the Gene Ontology at the Gene Ontology website. So why is Go useful? Well, it's useful in order to disambiguate language. So when we talk as humans, we are very good at interpreting things in a context. So if I'm a dentist and I talk about bud initiation, I might be talking about teeth, whereas a yeast researcher might talk about the budding that happens in cerevisia, or if you're a avid gardener, you might be talking about the new plants coming up in your garden. And similarly, you can also use different names for the same concept. So for example, the citric acid cycle is also known as the tricarboxylic acid cycle, TCAX cycle, or Krebs cycle. And I think different research groups or different fields of biology, they will adopt their own jargon. So in different fields, you might have these very different terms for exactly the same things. And it makes it very hard to compare across species, databases, and different research environments. So Go facilitates data aggregation for specific genes or individual genes. So it allows us to capture systematically gene-specific knowledge. So we are curating very detailed information and we annotate genes to these ontology terms. And it takes a long time. You have to search for literature, look in databases, do sequence analysis. But so far, across all these different annotations group that are part of the Go consortium, we have curated about 165,000 publications annotated for Go. And obviously, this is kind of data aggregation is not feasible for individual research groups to make these lists for every single gene of the 20,000 plus human genes, for example, or whichever organism you happen to work on. So in the Go, it allows us to regroup things. So instead of showing different attributes for genes on a gene by gene basis, we can make lists instead of genes with similar features. So for example, we can say that all these genes localize to the mitochondrion, or these genes are involved in DNA repair, or another set of genes comprise all the protein kinases in a cell. Probably won't be 2000, but yeah, just putting some numbers in there. And essentially, yes, we can group genes by similar features, which you can use to plan experiments, analyze data, and find trends in different data sets. What do they have in common? And as I already mentioned, the Go ontology consists of 3 different aspects or domains. So you have the molecular function aspect, and this refers to terms that describe single-step tasks of proteins or other gene products. So for example, you might have a gene that's annotated to the term protein kinase because it sticks phosphates onto other proteins, or it might ferry things around in the cell, or it might perform a specific catalytic activity. A second aspect of the Go ontology are the biological processes. And these are a recognized series of events. So for example, a pathway or a broad biological program, such as the citric acid cycle, cell division, or kidney development. And lastly, there's the cellular component aspect, which describes where different gene products localize within a cell or outside of a cell. So in the mitochondrion, in a membrane, or in a synapse, for instance. So a Go term consists of several different parts. It will always have an accession ID. Sometimes these parts will be referred to as slightly different things depending on in which resource you look at. So here I've called accession, but it might also be called, for example, Go ID. And they are stable. So they might be, so for example, I mentioned earlier that terms can merge, but then they will always become synonyms or still be associated with the new term. So they should be stable. There will also be a name. And when we annotate, we don't annotate to a name, but we actually annotate to the definition, which might be more specific than what the name implies, for instance. The term should also say what part of the ontology is, if it's a component, a process, or a function. And it will also specify relationships to other parts of the ontology, which I will go into more detail later on. Terms might also be associated with certain other attributes. So there might be tags and constraints that have to be followed when annotating to these terms. So for example, for the term mitochondrion, you can only annotate eukaryotes to that term. Here is another example, which is a function term. So GLO activity, or L-gluon or lactone oxidase activity. I think that's the enzyme that's mutated in humans, which means that we can't make vitamin C. But so this function term, it's an enzyme. So it also has, so in addition to all these other things I mentioned earlier, it also has cross-references to Metaseq and RIA databases. So the ontology is not a flat list. Terms are related to each other. And basically in ontologies, you can get different structures. So in a hierarchical ontology, you may have one-to-many relationships. So one term might have several descendant terms, but each child only has one parent. And that's not what the Go is. Go is structured as what's called a directed acyclic graph, or a DAG. In here, in this type of graph, you can have many-to-many parental relationships. So a term can have a lot of parents, and it can also have a lot of children. So why is it directed? So this is an actual example from the ontology. It's directed because you can only go up the ontology. you can't, it's acyclic, so you can't connect top terms to bottom terms. So for example, the mitochondrion is part of the cytoplasm, but the cytoplasm is not part of the mitochondrion. So here you see two different relationships. So you see this part of relationship and also an ISA relationship. So mitochondrion, for example, is an intercellular membrane-bounded organelle. The intermembrane space is a type of organelle envelope lumen and is part of the mitochondrial envelope. And what you see here in this graph is that when we annotate to this term here, mitochondrial intermembrane space, we also annotate to all the ancestor terms automatically. As a curator, I don't have to sit and annotate to each one on its own, thank God. And currently there are about 40,000 terms across the 3 different aspects in the ontology. So when you have a process, a broad, very broad program in a cell, for example, vesicle-mediated transport, this broad program can consist of several different ordered steps. So in this example here, I've shown vesicle fusion, uncoating, and docking, which are all part of vesicle-mediated transport. But these terms are also different because they are different, they're connected, even though they're all part of the same broad program, they can also be connected to other parts of the ontology separately on their own. So vesicle uncoating, for instance, has a path to protein depolymerization, and vesicle fusion is a type of organelle membrane fusion, which those other terms are not. So another type of relationship in the ontology is the regulates relationship. So I've shown ISA and part of. So you have regulates and also positively regulates and negatively regulates. So the term I'm showing here is positive regulation of MAP kinase cascade. And green positively regulates, so it positively regulates MAP kinase cascade. So you have all these links within the ontology, and the last relationship I'm going to show is the capable of relationship. So gene products or proteins may be annotated to respiratory chain complex 1, which is a cellular component. So cellular components, they don't actually comprise only parts of the cell, they also comprise complexes. And the respiratory chain complex 1 functions in the ETC, and this whole complex is capable of this molecular function. So a little bit more about annotation inheritance. So a very important feature of Go is that broader parents give rise to more specific children. And when you annotate a gene directly to a term, it's automatically annotated to all the parent terms. So if you search on a parent, you will retrieve all the children. So if you search for mitochondrion, you will also retrieve all the children that are annotated to mitochondrial lumen. But the other way around is not true. And therefore, every possible path from a term back to a root node must be biologically accurate, or the ontology has to be revised and tweaked. So functional biocreation. So basically, as biocurators, there are 2 main jobs we can do when we work with genomes. So we can either annotate features in a genome, so genes, promoters, and that sort of thing. But we can also do functional curation, where we associate these features, so for example, a gene, with different ontology terms. So what an annotation is, it's a statement that a gene product performs a specific function, or it localizes to a certain part of the cell or participates in some pathway. And currently in the Go, there are over 1 billion Go annotations. There are two main aspects to functional curation. So you get literature curation, which is mainly what I do nowadays, where you read full text publications and you associate the findings with the appropriate genes or features, sequence features. Not all curators curate on a gene by gene basis, some curate on sequence features. So like certain domains, for instance. And then you can also perform sequence analysis, yeah. So infer information from published genes. So manual curation is when different curators create annotations based on what's been published. And annotations can also be transferred to orthologist genes, either manually or automatically. And we also have electronic pipelines, which has given rise to the bulk of annotation that's been done to Go. And a lot of these are based on interpro signatures through a pipeline called interpro to Go, or are inferences made on one-to-one orthologs from ensemble gene trees. And these electronic annotations are not manually reviewed. And some people are disconcerted when they hear that, but it's been shown that these electronic annotations are very high quality, but they're very high level. So for instance, you might have a signature or a protein or a gene might match a signature for what a protein kinase or serine union kinase looks like, and therefore it gets that annotation, but it might not be as specific as the sort of annotations you can get when annotating a paper. It goes more into detail of the function. And obviously we can't just blindly transfer all annotations to all species, even if they are one-to-one orthologs. So for example, beak formation the chick BMP4 is annotated to beak formation. And obviously we can't transfer that to humans or mice because they don't form beaks. So when we make an annotation, we have to indicate what sort of evidence support the annotation, and there are lots of different types. They don't really indicate the quality of the annotation, although they might do in some ways, but they will give you a clue as to how the annotation was derived. So there's a number of experimental ones. That are from wet lab experiments. So you have IDA, which is short for inferred from direct assay. So these are things like a protein got GFP tagged and it was shown where it localized in a cell, or a catalytic enzyme assay, for instance, or IMP, which is inferred from mutant phenotype, when you show differences in phenotypes from two different alleles, or wild type versus mutant. There's a number of computational evidence codes. So ISO or ISS, when you infer things from sequence orthology or similarity. There's a phylogenetic code, IBA, inferred from biological aspect of ancestor. So where annotations have been transferred among related sequences based on common ancestry. We also have a no evidence evidence code, no evidence, inferred by curator IC. So that's, for example, if you have a protein that's been shown to be involved in transcription, you might be able to annotate it to nucleus, localize into the nucleus as well, based on an inference. We also have some author statements. If an author makes a statement in a paper saying this gene does this, and everyone knows that because it's been known for the last 70 years, but no one can find the experiment that shows that. And we also have the automatic code IEA. And the main difference between the automatic one and these other ones is that the automatic ones are made by a pipeline, and they are not, each annotation is not manually reviewed, whereas for all these other ones, a curator has looked at the annotation and decided to assign it. So in recent years, there's been some developments to provide more detail to the Go annotations. So the Go annotations are quite flat. There are these statements that this gene localizes to this part of the cell, or it does this function, or participates in this pathway, and there's no real connection between the different annotations. So the annotation extensions, they function partly to put gene targets onto a function, but also to connect different annotations to each other. So we might say for instance, that a kinase phosphorylates this other protein. So you can specify the other protein in an extension. Or happens during. So this biological process, stress-activated kinase cascade, happens during a response to osmotic stress. And these lend support to creating networks of annotations, connecting and stringing together the annotations. So a very new development, and even newer development at Go, is these causal activity models known as GOCAMs. There's a few thousand of them now, I believe. We don't do those models at Uniprot. But the idea of them is to provide a framework in which Go annotations can be linked into models of biological systems. So as I mentioned, a Go annotation is the association of a gene product with one of these with a term from one of these three different aspects. And there's been very little representation of how those genes fit together. So these GOCAMs were developed to try and enable pathway visualization. So in this example, we have NED4 that's been annotated to these different ontology terms. So it's a ubiquitin protein transferase, it binds RNA polymerase, it localizes to the nucleus, and it negatively regulates transcription, and is part of the cellular response to UV. So these are all very separate, disparate annotations. Whereas in a GOCAM model, you would say that this activity, so the ubiquitin protein transferase activity, is enabled by this gene product, and it acts on RNA polymerase 2, localizes to the nucleus during this response. And this whole activity model negatively regulates RNA polymerase activity. So it's causal. And this figure was taken from this paper here. So you can represent more complex statements. So whereas in the traditional Go annotation, you have an active entity, or what's called an active entity here, so a gene that you annotate to a term, here you start with an activity which is enabled by certain gene products. So it centers in a different way. So you have a molecular activity that's carried out by a gene product that acts on a different entity occurring in a location and part of a specific biological program, which could be part of a larger program or process, or for example, part of a certain cell type. So when you string several of these together, you start ending up with a network of different activities. So in order to access Go annotations, so you can download the Go annotations from, for example, the Go Gene Ontology website or from quickgo. There's also two main browsers for looking at annotations, and usually it's more user-friendly to look at annotations using browsers because they will display all these different relationships in the ontology. When you get when you download the ontology, it might not be obvious how all the relationships work. Different databases might also provide downloads to the data. And generally, you access Go as a GAF or in the GPAD format. So yeah, I mentioned quickgo and amigo. So quickgo is the EMBL-EBI browser. This is the link to it. I've also included links to a webinar that was done by George Yoru and a tutorial for it. So I'm not going to go into too much detail here since there's already a webinar on it. But I really like this browser for term browsing and looking at the ontology. Whereas amigo, which is developed by the Go, I quite like this ontology for accessing Go annotations because they have this super nice faceted search. Of course, you can filter down in quickgo as well. But I'm just going to show you quickly what I mean by this faceted search. So the main feature I like in quickgo is something called the ancestorview. So if you're interested to see how different ontology terms are related, if you go to the quickgo website and you start searching for terms, they have this little thing called a term basket that you can add your terms to, and you end up with this list of different terms. And then if you click this little button here from your basket, you can build these graphs showing you all these different relationships within terms you selected. quickgo will also show you co-occurring terms. So that's when a term has been co-annotated to other terms. I find it useful. So for example, so you can either show based on entire annotation set, which includes electronic annotations as well as manual annotations, or you can toggle to manual annotations only. So here I'm showing map kinase cascade. And we see for the manual annotations, map kinase cascade has most commonly been co-annotated, unsurprisingly, to map kinase activity, regulation of map kinase cascade, map kinase activity, and so on. So which all makes sense, but sometimes it gives you clues. I mentioned annotation extensions earlier. So quickgo also has a way of filtering for that using the filters they show on top. So if you load up all the annotations for a gene, they will have all these options for filtering things, and you can find the annotations. You can filter on annotation extensions as well. So this is CDC2, which is a homolog or ortholog of CDK1, and here you can see that it has got a lot of annotations to its target genes where it phosphorylates. And I mentioned amigo that has a nifty little species filter. So if you load up a term in amigo, it shows you the definition and so on, same for quickgo. And it has this little hidden link at the bottom showing a link to all genes and gene products annotated to the cell wall. So I searched for the term cell wall. And if you click on that, you get taken to a results page where it shows you all the genes annotated to cell wall. And it also has these filters on the left-hand side where you can filter down on your list. And I really like the organism filter because it allows you to, so if you expand it, you can see that, so the organisms that have a cell wall are plants, fungi, and bacteria. And if you click on the plus button, you can start filtering down. So here I've clicked on the plus button for fungi, and it started narrowing down. So we have 147 proteins in Aspergillus nidulans annotated to cell wall, 136 in Cerevisia, and so on. I find that quite useful when looking at different species. And there's a difference in the data that's found in amigo and quickgo, so be aware of this. Amigo takes a gene-centric worldview, whereas quickgo takes a protein-centric. So in UniProt, we annotate to proteins, whereas in the Gene Ontology, they will show annotations to genes. So obviously you might have several copies of the same genes. Oh yeah, and they also omit the majority of IAs due to the large size of the data set in amigo. So you will have more annotations in quickgo. So moving on to how you can use the gene ontology. So enrichment is a way to find, so if you have a list of genes, how do you find what terms are statistically overrepresented in your list? So basically what they have in common. So you compare the annotations to your gene list with the annotations in the entire gene set to see if you have any terms that are over or under representation compared to the background. So for example, if you have identified, if you have done a screen and you identify 100 genes, if you've identified 4 out of 5 genes that are annotated to galactose metabolism, this is significant. So enrichment provides you with enriched terms and a statistical measure of how likely your result set fall into that category by chance. So this is just an example of enrichment example from this paper where they challenged cells with different experimental conditions. So they challenged them to hydrogen peroxide and heat and looked at what genes are upregulated and downregulated. And they found, for example, that in hydrogen peroxide, genes that are involved in detoxification go up, whereas genes involved in translation processes go down. And to do an enrichment, you need 3 main components. So you need your set of Go annotations, and you also need a gene list of interest. So this could come from a phenotype screen or differentially expressed genes across different conditions, genes that interact with each other, or so on. And you also need a background gene set. And this is all the genes that you could have detected in your experiment. So you don't want to include genes in your background set that you could never have identified in your screen. So if you are identifying proteins, then you don't want to include all the non-coding RNAs in the background set. Or similarly, if you've done a microarray, and the array only has a certain subset of genes, then that subset is your background. So in your results, you get a long list of terms that are enriched. And you need to look at this list because not all of them will be physiological relevant. So if you have very unspecific terms, so the high-level terms, they're often quite uninformative for a physiological role. I'm just going to explain a little bit more about that. So, for example, this is an enrichment I saw in a paper, and they included things like, I mean, I'm not even going to look at the numbers here, but they included things like metabolic process, response to stimulus, and you wonder, like, biological regulation, and what does this mean? It doesn't really mean anything. Biological regulation, it could be regulating kidney development or respiration, and it's very different processes. Or response to stimulus, what kind of stimulus? A viral attack or hydrogen peroxide. And it's also useful to look for ancestor-child terms because you can get a lot of repetition. So, for example, this is the output, or a small part of the output of an enrichment, an example enrichment. So you see here that, for example, you have very similar terms that are very closely related in the ontology appearing. So we have 5 out of 16 genes annotated to ergosterol biosynthesis, ergosterol metabolic process, and so on. And similarly for alcohol metabolic process or alcohol biosynthetic process. So of course, when you are presenting your results, you want to narrow these lists down, so you don't show these sort of almost duplicates. So in your results, you should also see the number of genes that are annotated to the GO term in your list, and the number of genes annotated to the term in the background set. Yeah, so here we have, so it's 5 out of 16 genes in our list, but it's 5 out of 38 in the entire set of genes of 5,400. And the p-value is the probability of seeing at least X genes in the list of interest annotated to a specific GO term, considering how commonly it's annotated in the background set. And obviously, the smaller it is, the less likely it is to be observed by chance. So high-level terms, as I mentioned, are often quite uninformative for physiological role. So what did I mean by this? So very high-level terms will lump unrelated concepts. So here, the first, or the example I had earlier of an enrichment showed metabolic process. But actually, when you look at the ontology, metabolic process is such a high-level term that it's a little bit meaningless to show it as a result, because types of lipid metabolic processes include, for example, lipid metabolic process, carbohydrate metabolic process, DNA metabolic process, and mitochondrial translation, for instance. And these biological programs don't really overlap. So the genes that are involved in mitochondrial translation are very different from the ones that are involved in carbohydrate metabolism. There's no real overlap in those gene sets. So to lump those genes together with those ones in your enrichment results doesn't really mean anything. Yeah, I already mentioned that you can filter out the very similar terms. So this is an example where they did that. So they had a number of terms, obviously, in the enrichment, and they just highlighted the broad themes, the 4 broad themes that they saw in their data. So now I'm going to move on to talking about GO subsets or GO slims, which people often use for showing enrichment results. So what a GO slim is, and they're also known as GO subsets, is when you take a set of very specific annotations or detailed annotations and map them into broader, more high-level bins. And this is possible because terms are inherited, high-level terms are inherited when you annotate very specific terms. And this is useful when you want to visualize broad annotation categories for a gene list or an entire genome. So basically you have 3 different ways or common uses of SLIMs. So you can either make a whole genome SLIM in order to summarize an organism's biology. So this is useful if you want to plan curation, because you can see what areas are under-annotated, but perhaps also for research, to see where research efforts should be focused, because it enables you to identify, well, either unannotated or uncharacterized genes. And typically for those SLIMs, you want them to be biologically relevant, of course, but also not to have a lot of redundancy. And you want them to cover as many of your genes as possible, because if you have things in your genome that don't SLIM, you want to know whether it's because they are either unannotated or uncharacterized. Another way of using SLIMs is to provide a single gene overview. So for example, in a ribbon display, and this is an example from the Alliance website. In the new UniProt website, we're also going to show ribbon displays. So here, this is for TP53. And all the different GOSLIM categories that it's been annotated to. So the cell color indicates annotation volume to that category. And it doesn't really matter if these are overlapping in a way. And they would give you an indication as to what branches of GO are applicable to a gene. And yeah, as I've already touched on, the third main way of using GOSLIMs is to SLIM the results from an analysis to help you interpret your results. So from enrichment, for example. And for this, for data display purposes, you probably want to provide your readers with a small and comprehensible table to convey your message about your data. And you can get ready-made SLIMs at the GO site. And there are species-specific ones available, as well as a generic one. I know there's been some work on the generic GO set, and I'm not sure if it's been updated yet. But otherwise it will presumably be so in due course. But there's no real one-size-fits-all. If you're interested in a very particular part of biology, you might want to create your own SLIM with quite granular terms for a specific branch of GO. So there's this tool for making your own SLIM. You can also make them in quickgo. And there's also a script available from the GO consortium, map to SLIM. So some tips for slimming is to only SLIM one aspect at a time. You want to minimize the genes that don't SLIM. So are they unannotated? Are they unknown? Are they just not SLIMing because you need to include more terms in your SLIM? And you have to pick your terms manually. Unfortunately, there are no easily identifiable levels in the ontology. So you can't just have a cutoff and say beyond this specificity, I want to take terms from there. And yeah, you don't want to go too high because then you start hitting those biological uninformative terms that are useful for ontology purposes, but not so much for informing on biology. And you also might need to make decisions on whether to lump or split terms. So you might have a number of genes in your list that have a common parent that they're annotated to, but the common parent is not very biological informative. So examples of this are nuclear gene expression. The genes involved in that are very different from the ones that are involved in mitochondrial gene expression or vesicle-mediated transport versus nucleus cytoplasmic transport or metabolic process that I mentioned earlier. And this is a real example where it was shown how useful it is not to lump those concepts. So this was a phenotype screen done on a deletion collection in yeast in Pombe. And it was shown that for the deletion mutants that had a problem with spore development, they were annotated to cytoplasmic translation. Whereas for mitochondrial translation, they presented with the skittle phenotype instead, and they had no real problems with spore formation. And similarly for transmembrane transport and vesicle-mediated transport, most mutants or deletion mutants in transmembrane transport, they had no real phenotype. They were wild type. Whereas the ones that were involved in vesicle-mediated transport, they had these germination phenotypes. The problems and pitfalls, you need to look at your versioning. Sometimes in certain places you find shockingly old files, so don't download things from anywhere and look at when the files were created. So I've seen 10-year-old files hanging around, which is, yeah, as I showed you earlier, the ontology and annotations change daily. So check the timestamp and make sure you use up-to-date data sets. And not all genomes are comprehensively annotated. That's also something to bear in mind. Especially those genomes without any dedicated curators. And the unknowns are ignored by enrichment tools. You only enrich for, well, overrepresented terms. So if something isn't annotated to anything, these could be interesting too. So be aware of if you have any in your list. If you include certain evidence codes in your annotation set, be aware of what they are. They don't tend to be indicated for quality, but there are some computational prediction ones, which might give you more false positives. And don't exclude the electronic ones. They are accurate, just high level. And you want to make sure that you use the correct background set for enrichments. And remember for slimming that Go annotations are not mutually exclusive. Don't put them in a pie chart because it's not an annotation number total. Well, it is an annotation number total. It's not a gene product count. And yeah, you might need to look at the slim and include more terms. And there's some suggestions for further reading here. And there are lots of different tools to do enrichments. I would try different ones and compare the results and see that you get things that you expect and what the difference are. And as I mentioned, we have a large amount of unknown genes hanging about in the genomes of our favorite organisms. So in this paper, it was shown that for both the model organisms Cerevisia and Pombe, gene characterizations over time was very rapid up to the early 2000s. And after that, it sort of stagnated. So we only know just over roughly over 80% of proteins and genes do in terms of their broad biological role, like what pathways they act in. And then when they looked at human, it was shown that there was a very similar story in human, where only about just over 80%, 80–85% have been characterized. And remarkably, about 70% of all human research is on proteins that were known pre-genome sequencing. And if you have a genome with no GO annotation, GO recommends you to submit the transcriptome to NCBI, and then it will get these IAA annotations. They don't recommend you to create your own IAAs because there's a lot of accuracy concerns. And that's from the Gene Ontology FAQ. And for some further help and contact, so GO has a help desk for questions and feedback. They have an FAQ and documentation. And then there's some geeky things as well on GitHub and APIs and so on. But yeah.
So one question is, why is there a need for an alternative accession ID?
Oh, it's only because sometimes you might merge different terms. So I can't think of an example, but you might have two pathways that were named differently, or maybe we had a separate term for a small part of the pathway, and then it got merged into a larger pathway because it's more useful to think of the pathway as the larger thing, for instance. So that's why terms might end up with sort of synonymous terms, but they refer to the same concept now in the biology. If
Okay.
that makes sense.
Another interesting question is, if the automatic annotations are manually reviewed, how the consistency or reliability is maintained?
I presume that they mean if it's not manually reviewed, but we do manually review them to some extent. So we don't manually review them on an annotation by annotation basis. But when you look at individual genes and you might spot that it has odd annotations, or that an interpro domain is associated with a term it shouldn't be associated with as part of reviewing other annotations. So they do get looked at. It's just that not every single one gets looked at individually.
Okay. Okay, so next question I will read is, for many life scientists, the learning curve for adopting ontological semantics is very steep. So to motivate them, what is the best successfully adopted use case that can illustrate the benefit of using Go ontology? So another question is, do you recommend any tool for local Go term annotation, given a big data set and possibility to use a computing cluster, or for private data set that cannot be uploaded to online services? Is there any resource for these cases where the user prefers to download an annotation tool?
So I think there are commercial software that you can pay for and annotate your gene sets with. And I can't think of what they're called. I've never used them. I've always worked in the public domain. So I can't recommend a specific one. And all I can really say is that the Go consortium doesn't recommend that people annotate their own data sets.
How can we obtain statistical value for an identified Go pathway, say in a differentially expressed gene list?
So when you do an enrichment, you should get the statistics with it as well.
All right. So next question is, I've seen that string database offers a functional enrichment analysis with Go terms. How good is these analysis?
I don't know. I haven't used string for years, and I've never seen their functional enrichment analysis. Sorry.
Right. So next
string is nice for visualizing certain things, like visualizing interactors and things like that. But yeah, I have no idea about their enrichment tool. It's a very specific one. I really like the Princeton one. I find the Princeton one very user friendly, but it depends on what your data are, because some tools will allow you to upload your own gene sets and annotations, and some won't. So it depends on a little bit on what the data set is that you have, and so on.
Okay, I think given the time, we can just have one more question. If I would like to look for enriched terms among a panel of immuno-related genes, for example, how should I behave? Should I narrow down the Go terms to avoid an overexpression of immunosystem-related pathway, which I would expect given the panel?
Yeah, I think if you take out a list of immuno-related genes, you would expect to see immune system processes overrepresented. But I don't think you should filter your annotations before you start, but maybe they're not so interesting to show if you want to present your results. So for presentation, I suppose you would remove them.